Welcome back to Human Biological Sciences 2. Let's continue our discussion on the blood with a look at red blood cells. Red blood cells are quite small. They're only around about 8 microns in diameter. Some of the capillaries that red blood cells have to flow through are so small that red blood cells have to bend in order to get through, and they stack up a little bit like dinner bowls. They're a biconcave shape. This helps them to be able to bend, and it also increases their surface area to carry oxygen. They're very flexible, and as I've mentioned, because of this, they're able to bend and squeeze through very tiny little capillaries. They don't have any nucleus or other organelles, therefore they cannot undergo cell division. So their lifespan is finite. They'll only live for about 100 to 120 days, and then they'll be removed from circulation in the spleen or in the liver, dismantled, the components recycled, and some of those components will be reused to manufacture new red blood cells. So very little is wasted. New red blood cells are entering the bloodstream from the red bone marrow at around about 2 million per second. They have surface antigens, and of course they bind oxygen because red blood cells carry a lot of hemoglobin that enables the binding of oxygen to be transported through the bloodstream to the tissues. The normal red blood cell count in males is around about 5.4 million per cubic millimetre, thereabouts. Females, about 4.8, thereabouts, give or take. Of course, females, it can fluctuate significantly depending on their menstrual flow. New red blood cells, as I've mentioned, they enter circulation at 2 million per second. So if 2 million per second new ones are entering the bloodstream, it's assumed then that 2 million are dying and having to be broken down and recycled in the spleen. The hematocrit is the percentage of red blood cells in any given sample of blood, and it's usually around about 45%, but it again can vary between sexes. Females, it can range from 38%, which you might see in a female who perhaps has a more heavy menstrual flow, where a lot of red cells are being lost every month. In a male, it will be a little higher, of course, because males are bigger. They have bigger muscles that require more oxygen, so the red blood cell percentage is usually a bit higher for them, and it's increased also by testosterone. This image here also shows again the blood sample that's been put through a centrifuge, and the heavier erythrocytes or red blood cells fall to the bottom. So this represents about 45% of the entire blood sample. Now, hematocrit can change significantly in disease states. One condition that I'm sure you've probably all heard of is anemia. Now, anemia is really an umbrella term for any condition that reduces the oxygen carrying capacity of the blood. So there can be a number of different types of anemia that will impact on the red blood cell's ability to carry oxygen throughout the bloodstream. For instance, there may be iron deficiency anemia. Now, we know that iron is a very important component of red blood cells for carrying oxygen. It's part of the hemoglobin molecule. So if there's something that decreases the amount of iron available to manufacture red blood cells, then the red blood cells are not going to be as efficient, and there may not be as many red blood cells. But there can also be folate deficiency anemia. There can be B12 deficiency anemia. Or perhaps something's happened to the bone marrow, where the bone marrow is not producing enough red blood cells, a condition known perhaps aplastic anemia. Or sometimes red blood cells are taken out of circulation too frequently, or too many are being taken out of circulation and broken down, perhaps because they've been damaged in some way. I'm sure you're all aware that some people have to have heart valve replacements. Now, these are quite hard structures inside the heart, and with the enormous turbulence inside the heart and the pressures involved, some red blood cells can get damaged as they're circulating through the heart. So these red blood cells will immediately be detected by the spleen as being worn out or damaged, and will remove those red blood cells from circulation. Now this is happening continuously because the blood is continuously going through the heart, and some cells get damaged. So the bone marrow may not be able to keep up with supply. So this is a term that's called hemolytic anemia, where too many red blood cells are being destroyed and the body can't maintain adequate production. There's also sickle cell anemia. We'll talk a bit about that later. That's a genetic disorder where the cells take on a very odd shape. It's a sickle shape, and they don't function in transporting oxygen as well as a normal cell does. Then we can go to the other end of the scale, where there are too many red blood cells. Now this is called polycythemia. Polycythemia vera is a disease where the body is actually malfunctioning and making too many red blood cells. Now we might think, hey, great, that's fantastic. More red blood cells, more oxygen. It all sounds pretty good. Unfortunately, red cells are the primary cells in the blood that increase the viscosity or the thickness of the blood. Now remember I said that the tiny little capillaries, some of them are so small that red cells have to actually bend and stack up to get through. They're relying on the watery component, the plasma of the blood, to sort of help them through those tight spaces. If the amount of red cells increases, there's going to be more of those red cells stacked up in those tiny capillaries, and it's going to get very crowded in there and very hard for those red cells to get through. So an increase in the viscosity of blood because of high red cell count is not a good thing, and it can lead to all sorts of problems. There's also secondary polycythemia that is like an acquired polycythemia. Now this can happen as a natural response of the body to people living in higher altitudes or mountain climbers, but even some athletes will train in high altitude environments in order to stimulate their bone marrow to produce more red blood cells, and they do this just prior to an event, a major event, so they've got a little bit of oxygen, extra oxygen on board, but they are taking the risk that the red cells will be too plentiful and can clog up those tiny little capillaries. Polycythemia can cause dehydration. It can cause tissue hypoxia. Hypoxia is a word that's used to describe low oxygen concentration in the tissues. Now, obviously, if the red cells are getting stuck in these little capillaries, they're not going to be able to provide the tissues with the oxygen that they're carrying. So the oxygen, even though the red cell count is high, the tissues may still be low on oxygen because the cells just can't squeeze through those tiny little spaces. And blood doping by athletes, of course, this is very illegal. It can be detected by tests. Some blood doping takes the form of athletes having their own blood taken from them, and then they'll have that administered to them or transfused back to them just before an event. Now, of course, that's illegal. You can't do that in elite sports. The other way of blood doping is giving EPO or erythropoietin, which will stimulate the bone marrow to produce more red blood cells. Now, of course, that's illegal as well, and some athletes, or at least the people who advise athletes, got very clever, and they found a whole lot of drugs that were masking agents that wouldn't pick this up. But of course, then the Olympic committees got clever as well, and they manufactured tests that can detect these masking agents. So now athletes, not only can they not inject with EPO, but they better not be taking any of the masking agents either, because either way they'll be disqualified. So this is pretty much what I've been talking about in the previous slide, where the red blood cell count can be manipulated through various mechanisms, either by transfusing your own blood or your own red cells back into the bloodstream just before an event, or through receiving erythropoietin. In fact, one of the masking agents for erythropoietin is a diuretic called Lasix or frusemide. You may or may not have heard of that. So athletes are not allowed to take diuretics because that will be deemed to be a masking agent, and they'll be disqualified from events. Very dangerous to do this. As I mentioned, it increases blood viscosity. The poor old heart has to work a lot harder in order to get that thicker blood through the vessels. So the heart can become permanently damaged, a condition known as cardiac hypertrophy, where the heart gets bigger and bigger and bigger, the muscle hypertrophies, it gets bigger, and we might think, oh, a bigger heart, that's great, it can work harder, but in fact it may be bigger physically, but physiologically how it works is not as good. Now, hemoglobin, I've mentioned this a bit as we've been going along. It's made up of a heme and a globulin group, and it's the major component of the red blood cells that binds to oxygen. Each hemoglobin molecule can bind 4 oxygen molecules. And an interesting little bit here is that, remember I mentioned that if you were to prick your finger and put that little drop of blood onto the head of a pin, that would represent around about 1 cubic millimetre of blood. Now, in every cubic millimetre of blood, you'd have somewhere around about 3 to 5, averages out about 4 million red blood cells. Each one of those red blood cells will contain approximately 250 million hemoglobin molecules. Each of those hemoglobin molecules is able to bind 4 oxygen molecules. So that means every single one of those red blood cells can carry 1 billion oxygen molecules, and we have around about 4 million red blood cells on the head of that pin.
Hemoglobin is made up of a protein consisting of 4 polypeptide chains, which is the globulin component, or the globin component, and a heme component. One heme pigment attaches to each polypeptide chain, and each heme contains an ion that can combine reversibly with oxygen. Wouldn't be much good if hemoglobin bound to oxygen or oxygen bound to hemoglobin, and then hemoglobin wasn't able to offload that oxygen at the cells. So it has to be a reversible binding of oxygen. Heme is a ring-like non-protein pigment, and as mentioned, it's bound to each of the 4 chains. At the centre of each heme ring is an ion that can combine with the oxygen. Now, the hemoglobin function is to transport gases, but not only oxygen, carbon dioxide, it also transports nitric oxide, supernitric oxide, and also carbon monoxide, and I'll talk a bit about that in a minute. But where oxygen binds to the ion component of the hemoglobin, carbon dioxide binds to the amino acids in the globulin portion of the hemoglobin. Pretty much all of the oxygen that we inhale is transported through the blood bound to hemoglobin, about 98% anyway. Whereas carbon dioxide's dealt with a little differently. About 23% will be bound to hemoglobin, 70% will be in the form or transported in the form of bicarbonate ions, and there will be about 7% thereabouts that will be free in the blood solution. Nitric oxide is a very important substance that can help to fine-tune or manage our blood pressure. It is a pretty powerful vasoconstrictor. So in the event that our blood pressure would fall, then nitric oxide can cause our blood vessels to constrict, and that can help to increase our blood pressure. Whereas supernitric oxide does the reverse. It actually causes vasodilation. So in the event of our blood pressure going up very high, supernitric oxide can cause our blood vessels to dilate, and this can help to bring our blood pressure down again. Now, I said that I'd mention carbon monoxide, even though it's not on your slides. I think it's worth mentioning. Carbon monoxide is a gas that's produced when things are burnt. For instance, it's in car exhausts, it's in wood fires, it's in gas fires, all sorts of different combustion situations. When carbon monoxide is inhaled, it has a much, much greater affinity for hemoglobin than oxygen. So it will bind to that hemoglobin more readily than oxygen will. The problem is that carbon monoxide is very bad at disassociating from hemoglobin. So it's bound, not irreversibly, but it can remain bound to the hemoglobin molecule for a long, long time, effectively putting those red cells out of action, because while they've got carbon monoxide bound to them, they can't bind any oxygen. So that's why people can and do die very quickly from carbon monoxide poisoning, because it starves the tissues of the needed oxygen. Now, hemoglobin can vary, or the range of hemoglobin can vary depending on age and gender. And as you can see from the slide, infants, the hemoglobin is about 14 to 20 grams per 100 mils of blood. Adult females, about 12 to 16 grams per 100 mils of blood, but of course this can vary depending on the menstrual cycle. Adult males, about 13.5 to 18 grams per 100 mils. We've touched a little bit before on different types of anemia. Well, deficiency of certain nutrients has or can have a significant effect on the production of hemoglobin. One in particular is an iron deficiency, because I've mentioned before that iron is very important for hemoglobin. So if there's insufficient iron in circulation or being stored by the body, then that can result in not sufficient amounts of hemoglobin being produced. Production of abnormal hemoglobin can result in very serious blood disorders, one being thalassemia and the other sickle cell anemia. We'll talk about more those diseases, conditions a bit later on. Although it's probably worth mentioning at this time about sickle cell anemia. As I mentioned, it causes the cells to take on a very strange sickle, sort of half moon shape. Sometimes they look like they've got little barbs on the side. And in sickle cell anemia, these odd-shaped cells start to leak potassium out of the red blood cells. Now, an interesting thing about this is that people who suffer from malaria, well, at least people who live in an area of the globe where it's possible to contract malaria, often don't get malaria because the malaria is contracted through the mosquito, and the larva of the malaria and the parasites need that potassium inside the cells of red blood cells in order to live inside those red cells. But because sickle cells leak potassium, the malaria parasites can't survive there. So what we see in the equatorial regions of the planet, where we see a higher incidence of sickle cell anemia, we also can see a lower incidence of malaria. I'm not sure which one I'd rather have. They're both pretty horrible conditions. Sickle cell anemia is awful, and so is malaria. So it would be hard to choose which one you'd want to have. Now, there's also a blood test called hemoglobin A1C. Now, this is a blood test for glycated hemoglobin. Now, glycated hemoglobin is formed when glucose sugar attaches to hemoglobin in newly formed red blood cells, so as soon as they're formed in the red bone marrow. The higher the glucose level at the time that these red blood cells are formed, the more glycated hemoglobin is present. So it's another way that diabetics can have their glucose levels monitored. Now, I mentioned before that red blood cells have a finite lifespan. It's up to about 120 days, but it's around about 100 to 120 days. And when they wear out, they are detected as a worn out cell by the spleen, or at least macrophages inside the spleen and the liver. They're removed from circulation and they're broken down, and their components are recycled. This slide, which comes straight from your tutorial textbook, shows the lifespan and the recycling of our red blood cell. So macrophages in the spleen, liver, some in the bone marrow, phagocytose or break down the red blood cells. The globulin and the heme portions are torn apart. Globulin is broken down into amino acids and other proteins. The iron is removed from the heme. It then attaches to ferritin and then reattaches to transferrin. The transferrin then goes to the red bone marrow for the production of new red blood cells. The stimulant for the production of these new red blood cells is erythropoietin. Non-iron portion is converted to biliverdin and excreted in bile. In the large bowel, some bilirubin is converted to urobilin and excreted in the urine. So the next few slides are really covering what I've just mentioned. When red blood cells are detected as being old, worn out, damaged, they will be removed from circulation in the liver or the spleen, broken down by macrophages. The globulin portion is broken down to amino acids, and those are recycled. And the heme portion is split into iron and biliverdin. Biliverdin is the green pigment that is added to bile. Iron is an essential component of red blood cells, and the body is very efficient at recycling it. So the iron from broken down red blood cells is recycled and reused. The biliverdin is a green substance that's converted to bilirubin, and bilirubin is then secreted by the liver into bile. Bile is an emulsifier that breaks down fats. It doesn't enzymatically break them down. It dismantles the fats. It tears them apart so that from big sort of fat globules into smaller fat globules, so that the enzymes that break down fats are more able to um, digest them effectively. Some of this biliverdin is converted to urobilinogen, then stercobilin, and this is a substance that gives fecal matter its brown colour. This is done inside the large intestine. Some of it will be reabsorbed into the intestine or from the intestines back into the bloodstream. It's then converted to a yellow pigment called urobilin, and this is excreted in the urine, and that's what gives your urine its yellow tinge. Erythropoiesis, of course, is the term that's used to describe the production of red blood cells or the formation of red blood cells. This occurs in the red bone marrow mostly, as we know, but during embryonic development, the yolk sac begins the process, followed by the liver, and then the red bone marrow takes over. The main stimulus for the production of new red blood cells is hypoxia. Now, I think I might have mentioned in a previous slide that the kidneys produce the substance called erythropoietin that stimulates the red bone marrow to start making more red blood cells. The kidneys are perfectly placed to monitor the blood. So in the event of a lower oxygen carrying capacity of the blood, the kidneys will start revving up the production and secretion of erythropoietin, and this in turn will rev up the red bone marrow, telling it to produce more red cells. But of course, red cells don't just happen. They go through stages of differentiation and maturation. Now, you might remember back to the previous part of this lecture topic, where I talked about the pluripotent stem cells, and their first line of differentiation is into the myeloid stem cells or the lymphoid stem cells. Just forget about the lymphoid stem cells for a minute. We're concentrating on the myeloid line of differentiation. So the myeloid stem cells will ultimately give rise to red blood cells, but as I've mentioned, the red blood cells go through stages of maturation. It begins with the proerythroblast. This happens on day 1. It's then followed by the transformation to a basophilic erythroblast, and then a normoblast, and then a reticulocyte. Now, reticulocytes actually do leave the bone marrow and get into the bloodstream, but at this stage they are not fully functional red blood cells. They still contain some organelles. Most have um, gotten rid of their nucleus. All of these red blood cells start their life with a nucleus and organelles, but when they become fully mature red blood cells, they no longer have a nucleus and have virtually no organelle matter remaining in their within their membranes. The reticulocytes do take a little time to completely mature into a red blood cell. In the event of somebody hemorrhaging, losing a lot of blood quickly, of course the bone marrow is really going to rev up production of these red blood cells, so much so that more reticulocytes will find their way into the bloodstream. So if you took a blood sample from somebody who had a recent hemorrhage event, you would find that there would be a much, much higher percentage of circulating reticulocytes that have yet to become mature red cells. Now, hypoxia really is the main stimulus for the production of more red blood cells. And there are different types of hypoxia. Tissue hypoxia is where there is simply not enough oxygen getting to the cells. This can occur if people move to higher altitudes. For instance, if you all of a sudden decide, I think I might climb Mount Everest today, you start climbing up and up and up and up and up. The air is getting thinner and thinner and thinner and thinner with less oxygen, and your tissues will start to starve of oxygen. Or there could be anemia, any of those anemias that I mentioned before, sickle cell, B12 deficiency, iron deficiency, and so forth. Anything that reduces the oxygen carrying capacity of the blood will cause a tissue hypoxia. Or there may be circulatory problems. There may be absolutely nothing wrong with the amount of red blood cells that are circulating. But you may have a problem with your heart that's not able to pump that blood around. Or you may have a problem with your blood vessels that are not responding properly. They may be very stiff and hard and non-compliant, and the heart may not be able to work hard enough to get that blood and the oxygen around the body to the tissues. The kidney responds to hypoxia in producing more erythropoietin, which will stimulate the bone marrow to release more, mature and release more red blood cells. And it does this through speeding up the development of the pro-erythroblasts. And that in turn speeds up the maturation to the reticulocytes, and then of course the fully formed red blood cells. This is a little diagram of the feedback mechanisms that you would have seen many times. Feedback mechanisms operate in exactly the same way with the same components. There's always a stimulus that alters homeostasis. There is always receptors that detect this change. There will always be a control center that knows the correct balance, the set points, and computes what to do about this abnormal condition. And there are always effectors that are able to change the conditions to restore homeostasis. Reticulocyte count. Reticulites, as I've mentioned, are present in circulation, especially if there's hemorrhage. But in any given blood sample, there's around about 0.5 to 1.5% of the red blood cells circulating would be the reticulocytes that are yet to become fully mature. In an anemic person, we might find that there is a lower count of reticulocytes, and in conditions like leukemia, which is a blood cell cancer, there may be low counts of reticulocytes if there's nutritional deficiencies, or maybe the red bone marrow is just not responding to the erythropoietin that is being produced from the kidneys. A high reticulocyte count may indicate recent blood loss, as I mentioned before, where there's going to be a lot more immature cells released quickly into the bloodstream to try and boost the amount of red blood cells to compensate for those that have been lost in hemorrhage. Okay, that's it for part 2. So have another little break, and when we come back, we'll be looking at white blood cells.